Of all the extraterrestrial places where it ought to be easy to find evidence of living organisms, Mars surely leads the list. After all, in the past half-century, a veritable armada of spacecraft has flown to the Red Planet. Landers have set down at many places on the Martian surface, and as we write this, the Curiosity rover is climbing a geologically interesting mountain near the planet's equator. Surely by now we should have gotten a definitive answer to the question of whether or not life exists now or has existed in the past on this planet. Think again. The fact of the matter is that ever since 1976, when the Viking landers became the first human spacecraft to visit the Martian surface, a low-level debate has gone on in the scientific community about the evidence, or lack thereof, for the existence of life uncovered by these machines. It's hard to overemphasize the importance of this statement. Barring the development of something like Star Trek's fictional warp drive, we will never be able to explore any exoplanet the way we have explored Mars. If, after half a century of intense exploration, we still can't decide if there is, or was, life there, what hope do we have of answering that question for a planet light years away? The search for life on Mars can be described as an exercise in frustration. Time and again we have found things there that could be explained by the presence of life, only to realize that ordinary chemical reactions could equally well explain them. We are left with a collection of hints, but no definitive answers to our questions. As we said, it's frustrating. There were 2 Viking landers which were successfully deployed at different places on the Martian surface in 1976. Each lander contained 4 experiments whose goal was to detect evidence for life. An analysis using an instrument called a gas chromatograph mass spectrometer, designed to detect and identify different kinds of molecules. A gas exchange experiment in which water and nutrients were added to Martian soil, which was then monitored for evidence of biological activity. A prolytic release experiment in which Martian soil was exposed to gases containing carbon, then heated to look for evidence of photosynthesis. A labeled release experiment, which we discuss in detail next. The results from the first 3 experiments were unambiguous. They found no evidence for biological activity and, in fact, found no evidence of organic molecules at all. However, these experiments were designed on the assumption that life on Mars would have a metabolism similar to that of life on Earth, an assumption that may or may not be valid, as we pointed out in Chapter 3. They were also designed to sample only the topmost layers of Martian soil, not deeper than about an inch, 2.5 centimeters. The results from the labeled release experiments, though, garnered the most attention and triggered a half-century of debate. Here's how these experiments worked. A sample of soil scooped up from the surface was put into a chamber, and a mixture of water and nutrient molecules was added. These molecules had been made to contain a large number of carbon-14 atoms. Carbon-14 is a heavier cousin of the more common carbon-12. Carbon-14 undergoes the same chemical reactions as ordinary carbon-12, but is radioactive. Consequently, its presence in any sample is easy to detect. The logic of the experiment was simple. If there were microbes in the Martian soil, they would metabolize the nutrients and give off radioactive carbon dioxide, which would appear in the gas above the soil sample. And lo and behold, both landers reported the presence of gas that had the labeled carbon dioxide. Unfortunately, the euphoria that surrounded this announcement was not to last. When more nutrient was added to the chamber for second and third runs, no more radioactive carbon dioxide gas was seen. Had the original signal been due to microbes, scientists argued, their population should have grown and should have released more gas each time the nutrient solution was added. If that signal had been due to a non-biological chemical reaction in the Martian soil, however, the reacting chemicals would have been used up in the first injection, and no subsequent interactions would take place. This, of course, is exactly what scientists saw, and the general consensus, then and now, is that the Viking landers found no clear evidence for life on Mars. Furthermore, subsequent experiments discovered ways that ordinary chemical reactions in the Martian soil could have produced the carbon dioxide that was detected. This isn't the end of the story, though. Since 1976, a small but vocal group of scientists has argued that the Viking data, properly interpreted, did in fact establish the presence of microbial life on the Red Planet. At a major NASA conference on extraterrestrial life in 2016, for example, almost the entire question and answer period after one presentation was taken up by a spirited, and sometimes heated, discussion of the Viking results. But it hasn't been the results of the Viking experiments alone that have kept hopes for Martian life alive. As early as 1971, the Mariner 9 spacecraft, in orbit around Mars, sent back photos of the surface that looked for all the world like terrestrial river networks. Since then, orbiters and landers have produced incontrovertible evidence that liquid water once flowed on the planetary surface, and that early in Mars's history, its northern hemisphere supported an ocean. Since this would have been at the same time that life was developing on Earth, the idea that life might have appeared on early Mars gained a lot of traction. Even if that life went extinct when the planet lost its oceans and atmosphere, the argument went, we should be able to find fossil evidence for it. There is one characteristic of the Martian surface that has made scientists skeptical that evidence of past life in the form of organic molecules could persist today. Because it has no magnetic field, Mars is constantly bombarded by intense radiation from the sun. This creates high concentrations of hydrogen peroxide, H2O2, a powerful disinfectant. As a result, scientists believed that the Martian surface would, in effect, be disinfected, destroying any organic molecules created by living organisms in the past. In 2018, however, the Curiosity rover detected organic molecules in rocks that had been formed when there were still surface oceans on the Red Planet. And although these molecules were probably not created by living organisms, their presence gives us hope that molecules that were part of living systems there in the past might have persisted to the present. But what about life existing on Mars right now? We have sampled only the upper level of the planet's surface, reaching down at most a few inches. Could there be something important at greater depths? While the Curiosity rover was wending its slow way across the Martian terrain, the Mars Reconnaissance Orbiter discovered flow lines on the surface that darken with the seasons. These lines might be produced by occasional eruptions of briny water from the Martian interior, although some scientists have recently suggested that they are caused by flows of sand rather than water. In addition, in 2018, scientists analyzing data from the Mars Express orbiter suggested that there is a lake of liquid water under the Martian South Pole. And if there is liquid water under the surface today, it is reasonable to ask whether there might also be microbial life. This is one more possibility we have to consider. And then there is the methane. Methane is a simple molecule consisting of one carbon atom linked to about 4 hydrogen atoms. We know it as natural gas and use it to heat our homes and generate electricity. It is a minor constituent of Earth's atmosphere, constituting a little over 1,800 parts per billion by volume. That is about 0.00018% of the terrestrial atmosphere. Roughly 95% of terrestrial methane is produced by microbial biological processes, but there are non-biological processes that can produce it as well. When groundwater interacts with magma near volcanic vents, for example, or much more slowly, when ordinary chemical reactions in the environment convert iron oxide, rust, into certain other types of minerals. In 2003, astronomers on Earth, observing through telescopes, detected the presence of methane in Mars's atmosphere using a technique called spectroscopy, which we describe next. There wasn't much of it, only about 10 parts per billion by volume, much less than the concentration on Earth. But it was definitely there. Then, as the Curiosity rover made its way across the Martian surface in late 2013 and early 2014, something strange happened. The amount of methane suddenly shot up to 10 times the detection threshold, before dropping back down again after a couple of months. What could have caused this strange event, which scientists now refer to as a methane spike? It could have been the release into the atmosphere of a bubble of methane created by ordinary, non-biological reactions. Equally well, it could have been the result of a burst of growth of a population of subsurface microbes. While the existence of methane by itself is suggestive, it is definitely not proof of life underground on Mars. Another hint, another frustration. The Allen Hills are a God-forsaken expanse of Antarctica located about 130 miles, 200 kilometers, south of the main American base at McMurdo Sound. Anyone visiting the area will see only vast plains of ice, with glaciers slowly pushing up into a line of low hills. Most people's reaction to the place is simple. Why in the world would anyone want to go there? The answer turns out to be very simple. Meteorites. First, a word of explanation. When a meteorite lands somewhere in the glaciers around the Allen Hills, it embeds itself in the ice. As the glacier flows, it carries the meteorite with it. When the glacier is pushed up the low hills, the ice is worn away by the wind. The technical term for this is ablation, leaving the meteorite behind. You can think of the ice fields, then, as a kind of conveyor belt, catching meteorites and delivering them to the top of the ridgeline. In 1984, scientists riding snowmobiles across the glaciers picked up a meteorite. It didn't look impressive. It was the size of a grapefruit, weighed in at about 4 pounds, 1.8 kilograms, and was covered with the blackened coating that meteorites acquire as they streak through Earth's atmosphere. It was given the name ALH 84001, ALH for Allen Hills, and 84001 because it was the first meteorite found in 1984. Then, it was put in a drawer somewhere and forgotten for a decade. When it was finally analyzed in the mid-1990s, however, ALH 84001 proved to be a rare find. First, gas trapped in interior bubbles matched the chemical composition of the Martian atmosphere, showing that the meteorite had originated on that planet. This by itself isn't all that unusual. We have discovered well over 100 chunks of rock that were blasted off the Martian surface by asteroids and wound up on Earth. It was ALH 84001's timeline that really attracted attention. Radiometric dating established that the rock formed about 4 billion years ago, back when there was abundant liquid water on Mars. It was ejected from Mars about 17 million years ago by an impact, wandered in orbit around the Sun, and eventually landed in Antarctica about 13,000 years ago. ALH 84001, in other words, was formed at a time when life could have developed on Mars. It was a silent memorial to a period when our planetary neighbor was very much like Earth. In 1996, a group of NASA scientists led by the astronomer David McKay, 1936 to 2013, made an astonishing announcement. After examining ALH 84001, they argued that the meteorite contained fossils of living things that had existed on Mars. They based this claim on 4 findings: the existence of organic molecules called polycyclic aromatic hydrocarbons, PAHs, in the meteorite, the physical resemblance of mineral structures in the meteorite to fossils of terrestrial microbes, the similarity of combinations of minerals in the meteorite to those produced by terrestrial bacteria, the presence in the meteorite of chains of magnetite crystals like those found in some terrestrial microbes. It's hard to overstate the impact this claim had on scientists and the public. It even generated an announcement from the White House by President Bill Clinton, and it may have had a hand in creating NASA's current program in astrobiology. But as time went on, the inevitable counter-arguments began to surface. It was noted, for example, that molecules of PAH are ubiquitous in the universe and are found in many places, such as comets and even interstellar space, where there is no life. As far as the physical resemblance of the fossils was concerned, it was argued that the shapes of some known non-biological mineral formations on Earth mimic those of cells. In addition, the meteorite's claimed biological structures were about 100 times smaller than those found in any known cells on Earth. They would, in fact, have to be examples of a new class of life known as nanobacteria. Something that is theoretically possible, but has never been seen. Finally, it was suggested that some of the mineral combinations the scientists cited were the result of procedures used in preparing samples to be studied in electron microscopes. For a while, the magnetite chains remained the strongest argument in favor of the Martian fossil claim. Terrestrial bacteria use chains like these to differentiate up and down in murky pond water by following magnetic field lines. Since Mars may have had a magnetic field early in its life, it has none now, such an adaptation would make sense on the red planet. However, scientists have shown that the type of magnetite crystals found in ALH 84001 could have been produced by non-biological processes associated with the passage of the meteorite through Earth's atmosphere before it impacted on the surface. So, once again, we wind up with ambiguous and frustrating evidence. The structures in ALH 84001 could be Martian fossils, but they could also be the result of non-biological processes. Once again, we can't come to a definite conclusion about the current or past presence of life on the extraterrestrial planet we have explored most thoroughly. How then are we going to search for life on all of the exoplanets we know are out there? In the early years of the 19th century, the French philosopher Auguste Comte, 1798 to 1857, who founded a field that he called social physics and we call sociology, put together a list of scientific problems that would never be solved. A prominent inclusion on that list was the chemical composition of stars. Comte's reasoning was simple. In his day, the only way to discover the chemical composition of any material was to subject a piece of it to analysis in a laboratory. Since we can never get a chunk of star stuff on a laboratory bench, Comte argued, we'll never be able to find out what the star is made of. You can imagine him saying that since we cannot travel to an exoplanet, we can never know its chemical composition. In 1859, however, two German scientists, both known primarily for other advances, got together in a laboratory in Heidelberg and changed the way we analyze the universe. Gustav Kirchhoff, 1824 to 1877, is well known to physics students as the author of a set of laws that allow us to analyze complex electrical circuits, and Robert Bunsen, 1811 to 1899, invented the Bunsen burner, a fixture in every elementary chemistry lab. They introduced a process in which light from a heated sample of a pure material was passed through a glass prism to separate the colors. Instead of a continuous spectrum, like a rainbow, as they expected, they found that each chemical element produces a characteristic, unique, and well-defined set of specific colors. This collection is called an emission spectrum, and there is a corresponding spectrum associated with the absorption of photons of specific energies. This branch of science devoted to the study of these spectra is called spectroscopy. You are actually familiar with the fact that chemical elements emit specific colors of light. Have you ever noticed that some street lamps produce a yellowish light? Such sodium vapor lamps are often used in areas subject to fog because their color gives the best visibility in those conditions. Because each chemical element emits a characteristic set of colors, if we see that optical fingerprint in the light from any source, we can be sure that the source contains the corresponding chemical element. The point of this so-called spectroscopic analysis is that it makes no difference how far away the source of light is from the detector. It can be a few inches or billions of light years. Once the spectroscopic fingerprint has been created, it will stay in the light beam forever. One amusing sidelight. Today, a complex spectroscope can come with its own onboard computer and cost many thousands, even hundreds of thousands of dollars. Kirchhoff and Bunsen built the first spectroscope with a couple of old spyglasses and, believe it or not, a cigar box. It wasn't until early in the 20th century that the scientists who created the discipline called quantum mechanics finally discovered how atoms produce spectra. Here's a simplified picture of the atom that they developed. Unlike planets in a solar system, electrons in an atom cannot have orbits in arbitrary places. They can be found at only certain distances from the nucleus, in what are called allowed orbits. Each of these has a specific energy, so when an electron moves between them, the atom will emit or absorb a specific amount of radiation corresponding to the difference. It emits radiation if the electron moves toward the nucleus, and absorbs radiation if the electron moves farther away from the nucleus. Since the atoms of different chemical elements have unique arrangements of allowed orbits, each chemical element absorbs and emits a unique collection of radiation. This is what creates a spectrum. It isn't only atoms that emit spectra, however. Any system that can have different energy levels can generate a characteristic fingerprint. Complex molecules, for example, can vibrate, rotate, and change their geometrical configuration. Each of these modes gives rise to a characteristic spectrum. Thus, it would seem that the science of spectroscopy is an ideal way to search for molecules produced by living systems on distant exoplanets. Just find the signature of biological molecules in the spectra of exoplanets, and we will have produced incontrovertible evidence for the existence of life. Consider Earth. The chemical makeup of its atmosphere is heavily influenced by the presence of life. In fact, of all the many hundreds of known atmospheric gases, only a very few are not influenced by the presence of living things. Helium, for example, was formed in the Big Bang and makes up about 1% of the atmosphere. Argon is present in even smaller amounts, and it comes from the radioactive decay of potassium deep in Earth's interior. But otherwise, virtually every atmospheric gas is produced, destroyed, or modified by biology. The oxygen we breathe comes from photosynthesis, whereby plants use sunlight to convert water and carbon dioxide into carbohydrates. Ultraviolet light from the sun breaks up biological molecular oxygen, two atoms of oxygen bound tightly together, into single oxygen atoms. These then react with molecular oxygen to form ozone, O3. Respiration and the decay of organisms produce carbon dioxide, the reverse of photosynthesis. Other gases such as hydrogen sulfide are produced by blue-green algae, and certain types of bacteria give off methane, as noted. Life shows itself in the composition of Earth's atmosphere. We call these biologically produced chemicals biomarkers or biosignatures of life on Earth. One might think that it would be easy to simply look for such chemicals in the atmospheres of exoplanets using the technique of spectroscopy to determine if life is there. But there are three difficulties with this approach. The first difficulty is that the exoplanets are exceedingly faint to our view. We see them by the light they reflect from their central stars. Over the vast distances to even the stars nearest Earth, detecting the light reflected by a planet is incredibly difficult. Over the past few years, however, astronomers have used highly sensitive detectors, along with some rather clever strategies, to study the light reflected by many exoplanets. The most successful strategy is to measure the light of the star when the planet is behind it. Then measure their combined light when the exoplanet is in front of the star. Subtracting the former from the latter gives the light pattern from the exoplanet, what we refer to as its spectrum. The second difficulty is identifying the signature of specific molecules that are in the exoplanet's spectrum. As noted, each element and molecule has a unique light fingerprint. But most often, the unique characteristic in the light that identifies the biomarker is a very small portion of the exoplanet's total spectrum. This means we need to collect lots of light from the exoplanet, which usually requires large telescopes. The third difficulty is the most troublesome. How do we decide which biomarkers actually prove that life is present on an exoplanet? As we discussed, life produces or modifies most of the gases in the Earth's atmosphere, so it would appear straightforward to look for the same ones in the atmospheres of planets around distant stars to determine which show evidence of life. But as usual, it's not that easy. The problem is that non-biological processes can produce virtually every molecule that we consider a biomarker in Earth's atmosphere. Take oxygen, for example. Solar ultraviolet light breaks up water molecules in the atmosphere, releasing atoms of oxygen that can recombine to make molecular oxygen. So, although the vast majority of molecular oxygen comes from photosynthesis, not all of it does. Or consider methane. As we noted, it can be produced in a variety of ways, many not involving biology. The same can be said about hydrogen sulfide, which has a characteristic rotten egg smell, produced by sulfur-reducing bacteria that thrive in extreme environments on Earth, but also by volcanic processes. We could go on, but the point is clear. For just about every molecule that we could identify as a potential biomarker of life on an exoplanet, there is a non-biological production mechanism. Some scientists are thinking about using combinations of biologically produced molecules to establish the presence of life. Take oxygen and methane as an example. On Earth, the concentration of methane is not stable because it is oxidized, burned, in the atmosphere. Yet, it is clearly present because biology produces it quickly along with oxygen. If we turned off all biology on Earth, our atmosphere would lose its methane in just a few dozen years. Oxygen would stay around a few thousand years if all life were to cease, but it too would eventually disappear as it was incorporated into minerals. Thus, the presence of both oxygen and methane might serve as a biomarker, even though the presence of either gas taken individually would not. Finding biomarkers on exoplanets is certainly much more difficult than just looking for gases produced by biological processes on Earth. This is an ongoing area of research and debate within the community of exoplanet scientists. At the moment, the bottom line seems to be that we will not be able to establish an unquestionable claim for the detection of life by looking at the spectra of single atoms and molecules. At least, those atoms and molecules we can observe in the spectra of exoplanets. Searching for combinations of